Welcome to November's ECR Wednesday webinar, hosted by eLife, the series that aims to give early career researchers a platform to discuss issues important to you and your research career. Today, our speakers will share their thoughts about how to make science more reproducible. The webinar will begin with the panelists sharing their stories. Then, in the second half of the webinar, we'll be putting your questions to them. To ask a question, you can type in the question box on the GoToWebinar Functions panel, or you can tweet us. We are at eLife Community, using the ECR Wednesday hashtag. Please also join us on Twitter immediately after the webinar, where we'll be continuing the discussion under the ECR Wednesday hashtag. Finally, I'd just like to let you know that we are recording the webinar and we'll make it available on YouTube in the near future. Now I'll pass over to Benjamin to introduce the panelists.
Thank you, Emma. So thanks all for joining in for this ECR Wednesday webinar. Today we're going to talk about making science more reproducible. And so I recently joined the eLife Early Career Advisory Group. And eLife has a long-standing interest in supporting early career scientists in their daily life, but also improving the overall standing in the research community. And some of these aspects cover reproducibility and new innovations in publications, for example, to share code within publications. So I joined EGAC recently, and one of my initiatives was to talk or get reproducibility, or how to be reproducible, more out to research and early career research especially. And this is really rooted in a personal experience I had, because in a lab I worked as a postdoc, we could not reproduce some of these major findings of the lab, which took us like 2.5 years of our time, a lot of gray hairs or lost hairs, actually to recover from this and actually retract the paper at the end. And at the end we could actually publish the right story, but I think a lot of this could have been avoided if protocols, for example, would be more thoroughly written and passed on between labs members when one set of lab members leave and new lab members come in, and also the lab implemented some of the source later on. So this is really my personal motivation about reproducibility, and now as I'm a little bit more by computational, I think code sharing and so on is also a very important part of science, which doesn't really happen. And in general, methods could be far improved to be properly shared. And yeah, when I wrote my PhD or my papers, nobody cared about methods, even though science is all about methods. So it's a little funny. But I hope we can address some of these issues and points today, and I'm really happy that we have 4 distinguished panel members. And before I start introducing them, we have a small poll for everybody. So, and the poll is, what do you think is the biggest challenge to creating reproducible research? So number 1 would be code sharing. Number 2 would be detailed of methods. 3, lack of statistical powers. 4, standardization of resources, or any others you can come up with. So please submit, and we hope to get the answers to you within 20 minutes. So now, of course, reproducibility has been an issue for the last couple of years. Many wolf cries have been about it, but I think we wanted to more focus today not only why science is maybe not reproducible, but maybe also how we could teach people, or how actually people can be more reproducible at the same time, with a positive spin of on the whole story. So for this, we have this distinguished panel. I'm going to do a brief introduction to everyone before we start off with Lenny taking on. So Lenny Tittelman, hopefully I didn't slaughter your name, sorry, as co-founder of Protocols.io, this is a protocol sharing website, and I have to declare my conflict of interest here slightly, because I'm also an ambassador for Protocols.io. But Lenny has over a decade computational experimental biology experience. He was at UC Berkeley and MIT, and now for 3 years, prior to switching math and computer science to molecular biology, he was a database developer. And Lenny is a co-founder of Protocols.io, a protocol sharing website, has a strong passion for science and improving research efficiency through technology. Our next panel member is Semine Bassi. She's a professor of psychology at UC Davis. Ms. Semine is an editor of several psychology journals. She has been working on transparency, replicability in open science for several years. She's president and co-founder of the Society for the Improvement of Psychological Science. You can find the blog when you go to this webinar's webpage. The next panel member is Hilmar Lab. He's the Director of Informatics in Duke Center for Genomic Computational Biology, and he is part of the Tata Carpentry Steering Committee. Hilmar's research interests are in open, reproducible, reusable, interoperable software and data, in particular biological data, recorded in form of natural language description. He has a dual background in biology and computer science. He is experienced in developing various informatics tools. Resources extends over more than 2 decades, includes commercial applications, real-time scientific data and acquisition software, bioinformatic data integration, data exchange, and ontologies. And importantly, Hilmar is one of the co-founders of Data Carpentry and the Reproducible Science Curriculum. And our last distinguished panel member is Monica Driscoll. She's professor of molecular biology and biochemistry at Rutgers University. Monica's lab focuses on fundamental problems in the biology of aging, with an emphasis on neurodegeneration, molecular strategies for health span extension. In 2013, she became involved in an interlab study to investigate problems arising from the reproducibility of results from respected labs involved in aging research, and has recently co-authored a piece in Nature talking about this challenge. So thanks everyone for joining us. This is great you're here, and I would like to take off with Lenny, the co-founder of Protocols.io, to share his story of reproducibility and why it matters to him.
Thank you, Benjamin, and thank you for sharing your story about methods, which allows me to cut down the first 2 minutes of my part, because I had a very similar experience, 1.5 years as a postdoc, fixing a single step of a protocol that I was using on single cell RNA fish analysis. And at the end of that 1.5 years, I realized that this is not a new technique, it's correction of something that's been previously published in Nature Methods. And even though I called the inventor of the technique, who's a professor in UPenn, and shared this, there's no way to really get this information out, right? So I get no credit for the 1.5 years of correcting literally a single step of a very long protocol. And everybody else who's using it, that's the really devastating part, everybody else who's using it is either making the same mistake and getting completely misleading results in this quantitative single cell method, or has to rediscover what I know and spend 1 year or 2 figuring out the exact same thing and changing that one step. And this is something that I know would love to share, but there's no infrastructure for doing that. So this led in 2012 to this unhealthy obsession with creating a central open access, free to read, free to publish repository where we can quickly share this information and the people who need to know it automatically discover it. So that's Protocols.io. And what we learned rather quickly is that we were setting out to solve the secondary problem, which is keeping methods up to date, the protocols up to date. The first problem is what Benjamin mentioned, which is that when you look at a typical research paper, the materials and methods don't really include the protocols that you need to replicate the results. And eLife itself in January published the first papers from the Reproducibility project, Cancer Biology, which was looking into what are the causes and how well can we reproduce things and what are the causes when we can't. And one of the big takeaways was that we don't have the detailed recipes, right? We have materials and methods that are full of contact author for details, or we use the slightly modified version of a technique reported in another paper, and then good luck figuring out what was modified. So that's why we've been in the reproducibility space for a while. But having said that, despite the fact that I'm CEO of Protocols, I actually don't think that this is a new problem or new issue that we have. It's not something that's come up in the last 5 years. I think it's new visibility and attention to a problem that we've always had in science, right? It's not that 50 years ago everything was reproducible and now it's gone to the dogs and nothing works. I think it's an issue we've always had, and I'm personally actually quite positive that when I look at the efforts and things that many of the people here on the panel and all over the world, scores of people are paying attention to the issue, starting efforts. It's not just about methods, of course. It's about the code, the data, the reagents that we're using and making sure that we specify them and finding contamination. It's about the incentives. It's about training. There's so many different components to it. In a sense, it can feel overwhelming, but I feel pretty positive about it when I look forward and what's happening, when I look at what's been happening over the last few years, and when I look at what I think will be happening over the next 10 to 15 years. I think that now that we have the internet and the attention to the problem, we can do a lot better. I don't personally think we're in a new crisis of some sort, but I think we can do a lot better, and I'm really excited about the many efforts that I see that I think are taking us in that direction. And with that, I'll stop a minute early because I had a good lead in from Benjamin, and I like Q&A more.
Thank you very much, Lenny. That was great. Now we move on to Samin Wazir. She's Professor of Psychology at UC Davis.
Hi, everyone. So it's really fun to be here as a non-life scientist. I'm a social and behavioral scientist, a psychologist in particular. And yeah, I've always been interested in methods. My substantive research is on the accuracy of self-reports. So I look at how well people know their own personality and when we can trust self-reports of personality, when we should use other measures. So questioning kind of the validity of methods and findings has been something I've been doing for a while. And I got interested in reproducibility in particular because it hit my field super hard in 2011. So social and personality psychology was kind of one of the eyes of the storm of the reproducibility crisis. And we're still kind of dealing with that. It's still very much a major debate in our field. It comes up at every conference. It's kind of having very far-reaching impacts on journals and on training and hiring and all these things. So my interest in it is really at that level. That, I mean, maybe not coincidentally, because I'm a social scientist, I'm interested in the interpersonal angle and the incentive structure and the politics and the kind of reward system, the human side of it. So what are the human biases that led to the situation that we're in science, where we've maybe not been as transparent and open as we should be? I think, you know, Lenny, you mentioned that you think it's not necessarily a crisis, and I agree that it's not new. Maybe a more positive spin on it is it's a revolution, that we're finally, I think, trying to get better. And there's a lot of debate about terminology in psychology about, you know, is this a reproducibility crisis or not, and so on. And I came across a term recently where they called it a credibility revolution. I think it was from an economics paper. And I really like that framing, because I think one thing that kind of ties all the branches of this across all the fields is the issue of credibility. And so, you know, there's the open access part of it and the openness and transparency part of it, which is really important. But there's also the rigor and reproducibility part of it, which is, you know, we want to make sure both the process is transparent, but also that the findings we publish are solid and are likely to be repeatable and so on. And I think what both of those have in common is credibility. And really, this does go back to the very beginning roots of science. So I teach a very intro first year undergrad class on research methods in psychology. And I start with the Royal Society's motto, take no one's word, and the Mertonian norms of science, which emphasize openness and transparency and skepticism and disinterestedness. And I want to instill in them those values, but I also want to be honest with them about how well we've lived up to those values and ways in which we may not have lived up to those values, but are now trying to do better. So I see that a lot of these changes, a lot of this reform is about trying to live up to the things we've always said we do. We always said science is self-correcting. We always said, you know, the motto of the Royal Society is take no one's word. We ask society to trust us not because we're saints and we're trustworthy, but because we're willing to show our work, we're willing to be scrutinized and have people be skeptical of our findings and see what if our findings hold up in that skeptical environment. So so I think that one thing I would say, in preparing for this, I was thinking about what can I share about the experience of being in social psychology, where we've been kind of roiled by this debate for a long time. One of the things I want to emphasize is that I think that a lot of the kind of nuts and bolts workflow, sharing, all of the software that's been developed that's for the sake of increasing transparency and openness is great. And I want to emphasize it's also for the sake of increasing rigor and reproducibility. That transparency is great for its own sake, but ultimately what it's for, in my mind, is being able to evaluate and assess whether the findings are solid and hold up and whether we should have a lot of confidence in them, or being able to find problems and poke holes in it. So it's not necessarily inherently going to lead to, it's going to lead to more criticism, right? The more open and transparent we are, the more we're going to be able to find flaws and critique each other's work, which is a good thing for science, but it's not necessarily going to feel good. And it might also mean that the answers to our questions are less exciting when we are held to this higher standard. And that's something I think we're struggling with in psychology, is that we got kind of addicted to really eye-popping, counterintuitive results and having major discoveries all the time. And so now to get a job or to get tenure, you have to have a major discovery in your name. And as we're doing things more rigorously and more transparently, I think we're realizing that that's an unrealistic standard, that discoveries are rare and counterintuitive findings are even rarer. So I think that we're going to have to think about how to deal with that reality. And one of the big things I hope we can talk about today is how to change the incentive structure, especially since this chat is targeted at early career researchers. I think a really big concern is that those are the people that are going to take the hit. So we're asking new trainees to do things the reproducible way, which means slower and less bold claims and more cautious and so on, which is great. But unless journals start rewarding cautiously written papers and more circumscribed claims, it's going to be very hard for those people to compete with others who are willing to make grander claims and use less reproducible methods. So I think one important thing in all of this is we need the gatekeepers like journal editors and funders to pay attention to this and reward behavior and methods, practices that are more reproducible, more transparent, and so on. And then one other kind of aspect of that is rewarding the critics, rewarding the people who are doing the self-correction, who are pointing out the errors, and one of the things I've noticed in psychology, and I think this is true in other sciences too, is that we like to say that we're self-correcting, but then we penalize the people who do the self-correction. So there's a lot of kind of questioning of the motives of why would someone spend their time just pointing out flaws in other people's research that's not productive, or that's, they must be psychopaths or something. But I think that if we want society to treat us as credible on the basis of being self-correcting, being skeptical, submitting ourselves to criticism, then we have to welcome that criticism, both as individuals but also journals and so on that need to be open to publishing critiques, to scrutinizing the things that have already been published and so on. So I think that's one kind of culture change I'd like to see, and I'm not sure exactly how to bring that about. So I'll leave it there.
Thank you, Samin. It was great having a slide from a biological point of view, different perspective. So we move on to Hilma Lab, and Hilma is Director of Informatics, Duke Center for Genomics and Computational Biology, and Data Carpentry Steering, member of the Data Carpentry Steering Committee. Hilma.
Yeah, so a number of really good points have already been made, and important considerations have already been expressed here. So I guess I can drop some of the things that I have been meaning to say as to the importance of reproducibility for science as a whole, right? It's really a cornerstone. That's why it matters, and that's why it should matter to all of us. So let me maybe take a slightly different tack. And focus on where I'm coming from to this, and that's essentially from three directions. Number one is through my day job, which, as Benjamin just said, as a director of informatics for Duke University's basic genomic science research center, right? So in part that involves the care and feeding of high-performance computing hardware, but in part I also have the responsibility here for promoting and advancing good research computing practices, right? And so we've heard some perspectives here on the wet lab side, where I'm coming how I'm coming to this is on the computational side, right? How can we make the computational side of research, which figures huge in genomic science, how can we make that more reproducible? And how can we promulgate, how can we identify and promulgate good practices, right? So that's one perspective from which I'm coming. Another perspective, really, I'm also a researcher. I'm also a PI on federally funded research grants. I have a research program, and so I also realize directly that reproducibility is actually hard. It's often seems to run in the face of exploratory data exploration, and algorithm development, and so if you're feet deep or knee deep in a research project, reproducibility always comes after, often comes after the fact, right? You're at the stage of publishing your work, and then you ask the question, oh, how about allowing others, starting from reviewers and to future readers of the paper, how can we allow them to and enable them and make it, make it easy for them to reproduce the research that we've done here and the results that we've arrived at. The third perspective from which I'm coming from is really a long-standing passion and activity in open source software development. I've been in this field for probably about 25 years now, and I've been very active, especially in the realm of nurturing communities that have the know-how and venues for know-how exchange, knowledge exchange on good practices, and how to do things well, including computationally, right? Including especially in terms of practices that enhance reuse of each other's products, and practices therefore also that enhance reproducibility of each other's results and products. And so so that's, it's these three directions that due to which I'm personally interested. And perhaps one thing I can say a little bit more about here, in contrast to the other panels, is from the teaching perspective. So Benjamin mentioned that I'm one of the co-founders of Data Carpentry, and have been on the board of that organization since its founding. But I'm also one of the co-founders of the Reproducible Science curriculum, which has recently been adopted by Data Carpentry. So it's now become a Data Carpentry curriculum and a Data Carpentry lesson, and is being developed in collaboration with Data Carpentry. And so one of the things that, once you start thinking seriously about how do you teach reproducible practices for computational science to your fellow researchers, whether at a center or whether in others in your research projects. One thing that comes up pretty quickly is there's this bewildering array of tools in this space, and it's still growing. It's it's still evolving, and so it can be quite confusing for what you could do, what you should do, what's worth to do more than others. So often there's a tendency to forget some very principal things that are important, I think, to keep in mind and to also emphasize in teaching. And that's basic. For one, it's basic messaging, namely, it's not, reproducibility needs to be done really, really well, or you don't even bother, right? That's not true at all, right? There is this thing of this notion of good, better, best, right? So any attempt at improving the reproducibility of your computational science will pay off, right? Any attempt is better than none. And so every incremental improvement is therefore also better, right? Has positive consequences. And the other thing to focus on really is that there's there are important concepts and important approaches that carry through no matter whether the particular implementation or realization of these concepts and approaches that you're teaching apply only to a certain environment, right? There are certain things that you can only that are specific to R Markdown or that are specific to Jupyter Notebook, but there are things that carry through all of them, and it's very important to emphasize them, right? And that's, for example, things like literate programming, automation, right? Do not repeat yourself, automated testing, archival, open archival of data, software, and other research products, and so on and so forth. And it's really useful, I find, to focus on these things when you teach, and make sure that everybody understands that when we're looking at any specific implementation of those principles, it's only an implementation, and they're available in many implementations. I guess the last thing I want to say is, because it's come up a few times, I think reproducibility and reproducible science practices need to be strongly promoted in this, not as a not as a virtue. They are a virtue, but they need to be promoted through self-interest, right? It's making especially to early career researchers, making your research reproducible may give up some short-term gains, but the research career that you have in front of you is long enough that you'll be able to reap the long-term benefits of that many times over, right? For your own research, right? It's your future self that will be able to reproduce the science that you do now, and that will thank you over and over. And I'll leave it at that.
Thanks, Helmer. That was some great closing remarks. Now we move on to Monica, and she's a professor of molecular biology and biochemistry at Rutgers University. Thank you, Monica, for joining us.
Hi, thank you so much for having me. So I'm going to share a little bit of the personal experience kind of being in the trenches in a major reproducibility study. So I am a C. elegans geneticist. For people who aren't familiar with the animal, it's a small 959-celled little worm that's a very popular model organism, and it only lives for 21 days. So it's been very popular for the study of aging and longevity. Those studies have involved both genetics, but in particular drug interventions that can markedly extend the longevity of this animal. And they've been associated with some major controversies of reproducibility over time. In response to this, or at least in part in response to that, and in response to this effort on the part of the US NIH to improve kind of reproducible experimentation, they established something that's called the Centroiditis Intervention Testing Program. And basically there are two things about this that are really relevant to the discussion of reproducibility. First is that inherent in the design, so the basic idea is to screen for chemical interventions that promote longevity and healthy aging. So in terms of accomplishing reproducibility, what the idea is that 3 different laboratories are involved in this effort. So there's a lab of Patrick Phillips at the University of Oregon, there's a lab of Gordon Lithgow at the Buck Institute for Aging Research, and there's mine on the East Coast in New Jersey. And so the idea is that the experiment should run exactly the same at all 3 sites, and for any data reporting we have to, obviously compare our outcomes. The other component of this that is particularly important is that although this model organism that we use, Centroiditis elegans, is celebrated actually for its genetic uniformity, the underlying principle of this particular screen that's looking for reproducibility and robustness is that we actually use 22 different strains, like wild strains, as well as the standard lab strain. And that's to introduce genetic diversity that, kind of analogous to what you might encounter ultimately for humans in the population that you're trying to provide some drug intervention for anti-aging impact. So, so this started us. So this was a goal. We started testing 10 compounds that had been used in the and published in the literature as having effects on lifespan, maybe being controversial or whatever. So our job was to reproduce these studies in three different sites. And the first thing kind of speaks to the issue of protocols. It was sort of my job to just take these are 3 expert labs who do C. elegans lifespan studies. We all thought we really knew how to do lifespan studies, but when I took a look at the 3 basic protocols, as soon as you, from the different labs, as soon as you just scratch the surface, the differences among the different labs in what we were doing, the reagents we were using, our criteria, dead or alive, blah, 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 blah. It was stunning. Literally, it took us a year in order to get to a protocol that was very, that in the end is very reproducible. In that, the devil in the details sort of story, we had amazing conversations about caveats and censoring. The technicians on this team, and there were many, had literally still have to have a weekly 2-hour conversation to stay on the same page. But the writing of the protocols involved hours and hours and hours of very dedicated work. In the end, so then we did this for a while, and we published the outcome and analyzed it in detail. And there's a couple of very interesting things that happened. The first interesting and thing was actually a success. When we looked at the statistical analysis of where variation in lifespans could be attributed. So we looked at were there differences among the labs, within the lab, according to strain and genetics, or the investigator. What we found is that among the labs there was essentially 0 statistical variation in outcome. And so this was really was great and certainly gratifying after the terrific effort that was put into get uniform protocols. And of course, that I should mention that it's not only the protocols, but we share every single reagent. We do every single thing possible to be doing actually the same experiment. But we noticed another thing, and that was that there was variation in outcome within each lab. About 15% of the experimental variation could be attributed to, it just happened in the lab. And in fact, it could not be attributed to anything. It happened in all 3 labs to the same extent, didn't matter who the person was. So we think that we're learning that for this particular outcome that we're looking at, there's natural variation among, kind of its biology. And so that can be daunting in a way, but in fact, if you design your experiments properly, you can conduct enough repeats that you are not concerned that you've lost everything because of this natural flux in the baseline of the experiment. We also learn new things that we never ever would have noticed or believed if things were not going exactly the same in the same 3 different labs. We found some really interesting bimodal switching that suggests novel physiological states. I won't go into it because we're not talking about the science aspect, but there was new discovery because we had worked so hard to coordinate our efforts, and then we actually really believed them when we saw them, rather than say, oh, something was strange in the lab that day. And I guess the other thing that I thought that I should mention about the whole effort is that we did find that some published results weren't exactly reproducible. When we looked at the differences there, people, so this kind of gets back to this point, people do the same experiment in slightly different ways. Depending on when you introduce the animal to the drug, you could have a difference. And so a claim that that particular outcome wasn't reproducible really totally depended on the circumstances of the experiment, not at all, you know, the nature of the rigor of both independent studies, but that you, of course, you always have to be comparing apples to apples. I think that our study raises the question a little bit of, like, you know, how much effort is needed to replicate, you know, could we really rework all of, you know, science, or could we, or should we, to do this sort of an effort? Probably not, but there may be certain things, for example, these, you know, the drugs, preclinical drug screens and things like that, that would benefit from an analogous sort of organization. And then I don't know how I'm doing on my time, so stop me if I'm going over time. I guess I would say so having gone through this excruciating level of detail and planning on one project has actually, you know, filtered over onto other projects that I have going in the lab. And so we're working on we have, I don't know, some subtle phenotypes. And so we're much better understanding the communication of and coordination of both the experimental design, what's allowed, these little, like, you know, strange things. You know, how hot is the microscope when animals are sitting there? They're things that you become aware of, and it is good to record them. The other thing, what was I going to say about that? Oh, yeah, so I may kind of leave it at that, but in terms of teaching within a lab, this rigor and reproducibility, I agree that it's your, you know, it's your own scientific integrity. You really want to get it right, and to get it right, you really need to be able to tell people exactly what you've done and do it in a sensible way. In addition, what I try to do is have people work more now than in the old days in groups. So one investigator, they're in a team, and so they blind experiments for each other. They you know, like, if person, you know, researcher 1 says A, then I have ask researcher B to do that again, you know, to go off and do it. And so there are ways that I think that you can introduce into your own lab this kind of reinforcement and reproducibility, which helps things go faster. And so I'll stop there. Happy to discuss anything else more.
Thank you very much, Monica. That was great. Reinforcing this reproducibility just also leads to new discoveries. So I think that's really important to notice this as well. So before we actually open the floor for questions or discussions between the panel members, we have our results from our poll. And so it was, what do you think is the biggest challenge to creating reproducible research? And I guess the big lead really was the detailed methods, which obviously, as Monica just pointed out, actually writing good methods is a really hard deal, because, like, there's so many small little factors which can influence an outcome. And I guess it's also the same for coding. Like, if you don't really share your code, then how does anybody know what you actually did? I think that's pretty much the same in both areas. So do we have any questions for now? If not, we have a couple of prepared ones. All right. So I think what one thing I would like to go on with was really that reproducibility or credibility, how Simin really put it. I think I like this too, this credibility revolution. There needs to be an incentive structure, and I was wondering, and then Helmer picked it up, that the incentive structure could be also just for your future self, like maybe you comment a line in a code and then you actually know what your code did. The same as like how you actually did an experiment. But how do you think, and maybe I just opened this as a floor really here, how do you think that funders or journals or so on actually could provide a better incentive structure for reproducibility? So should open and reproducible science be part of like actually promotion or actually hiring processes? And how could this be implemented?
So, you know, one thing that I've wondered about, I mean, you are asking in a certain way for this profound restructuring of what we consider the standard criteria at the top tier journals. But so I wonder if in fact there could be journals or sections that are kind of dedicated to reproducibility studies and willing to publish both positive and negative sorts of outcomes. And so I think young researchers, you know, get some credit for any publication, you know, any publication that they get. And I personally won't even look at these like number rankings of, yeah. But so I wonder if you know, even each major journal was having a reproducibility section, and if the community gave that some sort of a, you know, a positive, you know, stigma, maybe that would be something useful.
All right. Well, we have to try to fix this technical glitch. There was an interesting question from one of our audience. So one of the questions was, how did the scientific community allow methods not to matter? Anyone of the, like, how did we actually get here? Any comment on this?
You know, maybe being one of the older members of the panel, I can address that. My impression is, so in the old days, there was no online journal. The journals were published as, you know, magazine-type units, and the, you know, there just wasn't a lot of space. And you know, in all honesty, I felt like, you know, publishing a Nature paper, which the wording had to be compacted, the methods had to be cut down to nothing to fill in these page limits, you know, really made it that you weren't giving the true representation of the overall body of work. Now, the journals have really done a lot, I think, to allow these enhanced method sections in terms of volume. But I do question, how would I say it? It's like the standard, well, the standard of statistics, I think, has gone up, but not necessarily methods. Like I feel there's very little checking on how precisely those methods are written, and I find in many articles now, you know, I wanted to know what was the source of that reagent, and this stuff isn't there even in an expanded thing. So I wonder. So just to answer the question, historically, I think the space constraints of publication caused us to devalue the working of the methods. I also think that probably we weren't as critical as maybe we didn't you know, it wasn't in our psyche as much how important it was to get those details out there.
Please, Danny.
Just a few notes to follow up on that. Regarding the poll, I actually disagree that methods are the most important thing. Even within biomedical, I think they're a component of the difficulties that we have with reproducing, and obviously I care about them a lot and have been obsessed with this for quite a long time. But it is many things, just as we heard in this entire panel. It's not that there is one key thing that's going to solve it, right? And we need the funders, the journals, the researchers themselves. There's a lot of players and a lot of us that can improve it. And as I said, it's the code, it's the reagents, it's the data. And as far as why we are where we are in terms of methods, it's what Monica said, but also if you think about a research paper, 95% of people, and I actually have statistics on this, who read a paper, they don't need a full 10-page step-by-step take out the cells. They want an abstract in the method. They want to get, when they're reviewing or when they're preparing for journal club, they want a quick idea of what was done. You got DNA out of an organism, you sequenced it, you use software to align it. It's the 5% of people that are going to reproduce it, replicate it in the same lab or outside that care about the protocol. And there wasn't a good balance. So if you had a paper, if every paper always included 10 pages of the step-by-step protocols, you wouldn't be able to read and get a gist of what was done. You need abstract, right? So we just didn't have the internet, we didn't have the capacity. We used to publish Excel spreadsheets as part of our papers with the sequences, right, with the genome sequences. And we're now at a point where we have Figshare and Data Dryad, where we have Jupyter Notebooks and CodeOcean and Protocols IO. And so I think one of the big things that's happening is the paper is moving away from the PDF, and we are now finally at a point where we can use the internet to create an interconnected sort of research output where you have the PDF, which is the story, connecting to the code and the data and the methods, and you know, linking to the AdGene repository so you can get the plasmid. And that's really why I'm optimistic. There are some things that we can improve, even if we can't get NIH to do something about training, or journals to do something about checking whether the methods are correct, because that's really hard to do.
I'm not in disagreement with anything that's been said here, but I think it's worth saying something that hasn't been said yet. And that's, you know, personally, I think in a nutshell, the reason we're at where we're at is because of super competition. And, you know, if you look at, so it's true that there's been this transition from traditional paper-based publishing that has strong space constraints and, you know, costs that scaled pretty much linearly with the space occupied. And we've had this transition to all online, right, where really those cost, you know, those cost relationships should fall away. That having said that, if you go back to you know, scientific journal articles from, say, the end of the 19th century or early in the 20th century, they, you know, which were all print-based, right? There wasn't even any halfway online dissemination of research back then. They described their methods in very meticulously. Every single detail is there. There's also every single detail about the data. Every single data point that was obtained in the articles that I read from that time is in the paper. It's not in some supplements that you can't get to. It's not hidden away. It's not concealed. It's all there. And so I think, you know, one, if not the elephant in the room here is this super competition state of the field that we're in, where what's now matters more than anything else is having the discovery on your record. Whether that discovery holds up as reproducible, as science that's reproducible, or does not, almost takes a backseat. And I think, you know, one thing that needs to happen is to come back to the foundations of science. You know, that discoveries are only scientific discoveries once they hold up to scrutiny and to reproducibility. And that costs time and effort.
Right, so we have a question from Monica. So the question is, I read it out for you. I'm not an experimental biologist, but do you think that multiple people repeating the same experiment within a lab has been avoided due to authorship concerns? Also, how do you handle authorship concerns if multiple people work on the same project?
Yeah, so that's a good question. Just in terms of practical authorship order, one nice thing, of course, now journals will let people be co-level authors, and I think that helps. Among our group of 3 PIs, we just agreed, you know, we're kind of more senior, but we just agreed we'll just cycle it around, maybe according to more effort, but just equal among the 3. So it's a very cooperative, nice interaction. So the key point of your question was, wait, senior moment here. You wanted to know
multiple people doing the same thing.
How do you handle it? Yeah.
Right. So multiple people doing the same thing is you know, it's very hard to have that sort of structure in the modern world, and I'm actually not sure you absolutely have to. For this particular project, it had an emphasis on, or has an emphasis on, the reproducibility as really helping to jettison these compounds forward in the pipeline, and something that for a smaller organism is kind of tractable. But people doing the, you know, a couple people doing the same experiment is, you know, it's not the normal thing that we do. I agree. I think it's probably appropriate for things that are going to require a heavy investment, and then probably the slower path towards reproducibility, the more traditional one that is science practice right now, is that we would publish something, you know, not from this study, but something else, and share reagents, and when our colleagues in the field are building on that, part of the building will depend on whether the premise, the foundational work is reproducible. And so, yeah, I can't, you know, we can imagine a world in which, yes, there were sets of 3 labs doing all the same thing. We would have an increased rigor and reproducibility. That, I think, is not a practical solution, though.
So we're going to wrap up soon, but Simin, are you...
Can you guys hear me?
Audible? Ah, yeah. Maybe like we get the last 2-3 minutes to you because you were like off and on. So,
sorry about that. So I missed the last few minutes because I was restarting the computer, but I want to go back to something that came up at the beginning about why we've been neglecting methods and what could journals do to improve reproducibility. And Monica, I think you mentioned, you know, journals could maybe have a section for reproducibility or a separate journal. And I think that part of the problem is that we've accepted this kind of incentive structure where journals are motivated to accept and publish the most eye-catching stuff. Their main incentive is to increase their impact factor. And I know a lot of people say that we don't care about impact factors, but that still seems to be what drives a lot of decision-making and bottom-line thinking at journals. And I mean, ideally, the editor wouldn't be worried about that. There's some separate committee that's, you know, worried about the financial viability of the journal and so on. But in reality, we're human and these things influence us. So I think we need there to be an actual, maybe even financial, but some very concrete incentive for journals to care about more than just how much impact a paper is going to have, how many eyeballs or, you know, clickbait or things like that. And so one of those things is things like replication and verification and reanalysis of results and so on, to make sure so that if journals are publishing things that turn out not to hold up, not to be reproducible, that there's some consequence for them. And I think this shouldn't be just one part of journals or one, you know, a subset of journals, but I think the most prestigious journals should be held to the highest standards from this perspective. I think part of why we got away from providing details of methods and providing code and data and so on is because you can get away with making bolder claims the less transparent you are, the less. And so the incentives were kind of aligned for the authors and for the journals to do the thing that will get the most attention, the most, and not consciously. I don't think people knew that they were exaggerating necessarily, but there was no break system on that. It was, well, we can claim this, we can cherry pick this result, present it. The journal likes it better that way. We like it better that way. So we need some kind of system to counteract that force for getting the most eye-catching results.
We have maybe like time for one more reply. Lenny, you had something. Do you want to say something?
Yeah, just very quickly. It was actually a question for Simin, where she said that in her introduction, she said that reproducibility is hard, which I completely agree, but also being transparent opens us up for more criticism, for more people finding fault. But on the other hand, my question to Simin is, isn't that counterbalanced by the fact that if you publish in a way where your method is more reproducible, you'll get more citations, you'll have more credibility in the field. There are also incentives if it's not on your hard drive, the data set and the scripts, but is on GitHub and on Data Dryad. 5 years later, when people email you about your paper asking you, can I get the code? And you're like, crap, I threw out the computer. It's really nice for you. It's really nice within your lab, like Benjamin was saying, if we have the details, if we have it all in the infrastructure that we have now with the repositories. So on the other hand, there are incentives for the researcher himself to be more reproducible for their future self and for their lab and for the community. So there are also rewards. So my question is, the pro and con on the bigger scale, does reproducibility win out with the incentives?
I mean, I agree with you and with Hilmar about the long term. In the long term, I think you'll be better off and you'll earn more respect and more credibility by doing things in a reproducible way. The question is, will those early career researchers survive to reap those benefits? And that depends on the structure of the field and how much people are attracted to glamorous, sexy results. And if there's too much of that, if people are too enamored with, very eye-catching results that without verification, without requiring that they post their materials and post their data and be held to a lot of criticism and so on, then the people doing the longer, slower thing won't be around to reap those benefits. So I think we need to balance it. We need to create that incentive structure, both by rewarding people who are doing these slower, more reproducible things, but also by resisting the urge to buy into these very appealing, eye-catching things unless they have all the characteristics of reproducible research.
Cool, thank you. That was great. That was also leads me nicely into actually wrapping this all up. I think it was a great talk, and I think thanks for everyone for joining us here. I think there was many points raised, and for a start, really the super competition we're in and this eye-catchy research we need to do. I think reproducibility just was an add-on and not really the foundation, while credibility was always part of research, and actually being questioned about your research should be part of getting better, and that's what science should be about. And as Hilmar pointed out, there's no holding back in getting more reproducible or more keeping more track of your research. So every little helps. He put it in it as good, better, best practices, but don't shy away to starting it just because you can't be as reproducible as you would like to be from the start. And then really that we need incentive structure, so to actually promote reproducible research. And this is once, of course, your future self going to be really happy about having proper methods and good documented code and code out there and data out there. But at the same time, you have to survive actually to reap your benefits. And I think that's a very important point to make, that, reproducibility takes time, and how is it valued, and how do we actually generate the right structures for funding or hiring bodies. So thank you again, Simin, Monica, Lenny, and Hilmar for being part of this. I think it was great. We're going to discuss a little bit more on Twitter when you have time or not. And thanks for all the listeners. Thank you.
Thank you for organizing.
Thanks.
Thank you. It was a pleasure to be here.